Welcome to G Fun Facts Online. Every year, millions of us get sick with the flu. But have you ever wondered how we fight back against an enemy we can't even see? For over 70 years, a silent, global army of scientists has been tracking the influenza virus, moment by moment. This is the story of that unseen sentinel network, our planet's primary defense against one of humanity's oldest microbial foes. To understand the fight, you have to understand the enemy. Influenza isn't one virus, it's a family. We mostly worry about types A and B. Influenza B causes a lot of seasonal flu, but Influenza A is the real monster. It infects birds, pigs, and humans, and is the only type known to cause devastating pandemics. The secret to its success lies in two proteins on its surface, hemagglutinin, or HA, and neuraminidase, or NA. Think of HA as the key the virus uses to get into our cells, and NA as the tool it uses to break out and spread. The virus constantly changes these keys and tools using two main strategies. The first is called antigenic drift. This is a slow, continuous process of mutation. As the virus copies itself, tiny errors creep into the genes for HA and NA. Over time, these small changes accumulate, altering the proteins just enough that our immune system, trained by last year's infection or vaccine, might not recognize it anymore. This is why we need a new flu shot every single year. It's a constant arms race. But then there's the second strategy, which is far more dangerous: antigenic shift. This is a dramatic, abrupt change. It happens when an influenza A virus gets a completely new HA or NA protein. Imagine a pig getting infected with a human flu virus and a bird flu virus at the same time. Inside that pig, the viruses can swap their genetic material. This can create a new hybrid virus, one that might have the easy spread in humans feature of a human virus, but with a bird virus HA protein that our immune systems have never seen before. This is what causes pandemics. The 1918 Spanish flu, the 1957 Asian flu, and the 2009 swine flu were all caused by antigenic shifts. Faced with such an unpredictable enemy, no single country can stand alone. That's why, in 1952, the World Health Organization formally established a global network to track the flu. Today, it's called the Global Influenza Surveillance and Response System, or GISRS. It's a massive collaboration with over 150 labs in 130 countries. Think of it as a multi-layered defense system. The backbone of this system is the National Influenza Centers, or NICs. These are the frontline troops. They're national labs that collect respiratory swabs from patients with flu-like symptoms. They do the initial analysis, figuring out if it's influenza A or B, and then ship a representative sample of these viruses up the chain. The next level up is the WHO Collaborating Centers, or CCs. These are a handful of the world's leading virology labs in places like Atlanta, London, Melbourne, Tokyo, and Beijing. They are the central command. They receive thousands of virus samples from the NICs and perform highly detailed analysis to figure out exactly how the virus is changing. This structure ensures that a virus from a single patient's swab can be detected, analyzed, and the data shared globally to protect everyone. So how does this molecular detective work actually happen? It starts with a nasal or throat swab from a sick patient. This swab goes to their country's National Influenza Center. The first step is usually a test called RT-PCR, which can rapidly detect the virus genetic material and identify its type. But to really study it, they need to grow the live virus, often in cell cultures or chicken eggs. Once they have a live sample, they ship it to one of the major WHO collaborating centers. This is where the really deep science begins. To see how much the virus has drifted, scientists perform something called a hemagglutination inhibition assay, or HAI. It sounds complex, but the idea is simple. They mix the new virus with antibodies taken from ferrets that were infected with last year's flu strains. Ferrets are used because their immune response is very similar to ours. If the old antibodies can block the new virus, it means the virus hasn't changed much. But if they can't, it's a clear signal that the virus has drifted, and our existing immunity might not work against it. Scientists use the results from thousands of these tests to create antigenic maps, which visually plot how different flu strains are from each other. It's like watching the virus evolve in real time. In parallel with this, they also perform whole genome sequencing. This means they read the virus' entire genetic code, letter by letter. This gives them an incredibly detailed view, allowing them to pinpoint specific mutations, identify dangerous reassortment events, and even track how a specific version of the virus is spreading around the globe. All this data is then uploaded to a WHO database called FluNet, creating a near real-time global map of where different flu viruses are circulating. This enormous global effort isn't just for science's sake. It leads to direct actions that save lives. Its most critical job is to help decide which strains go into the next seasonal flu vaccine. This is a high-stakes prediction. It takes at least 6 months to produce a flu vaccine, so experts have to analyze the viruses circulating now and predict which ones will be dominant half a year in the future. Twice a year, once in February for the Northern Hemisphere and once in September for the Southern, the WHO convenes a meeting of the world's top influenza experts. They pore over all the virological, antigenic, genetic, and epidemiological data to make their recommendation. Based on their expert consensus, they choose one AH1N1 virus, one AH3N2 virus, and 1 or 2 influenza B viruses to be included in the next vaccine. Another key function is monitoring antiviral resistance. Drugs like oseltamivir, known as Tamiflu, are a crucial second line of defense. But viruses can evolve to resist these drugs. GISRS constantly tests virus samples to see if they're still susceptible, ensuring that doctors have up-to-date guidance on which treatments will actually work. And perhaps most dramatically, GISRS acts as a global tripwire for pandemics. The system is designed so that any national lab that finds an influenza A virus it can't identify must immediately send it to a WHO collaborating center. The detection of a novel virus, especially one that has jumped from animals to humans, triggers an international alert. Scientists perform an immediate risk assessment, and if the threat is serious, they begin developing a candidate vaccine virus. This is a pre-made vaccine seed that can be handed to manufacturers to start mass-producing a pandemic vaccine at a moment's notice. This early warning system buys the world precious time. Despite its success, the system isn't perfect. Some parts of the world have surveillance gaps, creating blind spots. And the COVID-19 pandemic actually created a strange problem. With everyone masking and social distancing, the flu almost disappeared for a while, which made it incredibly hard to find enough virus samples to accurately predict the next season. But the system is evolving. The future of surveillance is integration. The COVID-19 pandemic showed how the flu network could be rapidly adapted for other threats. The new vision is a system that watches for influenza, RSV, coronaviruses, and other major respiratory threats all at once. Technology is also driving change. Next-generation sequencing is becoming cheaper and faster, allowing for real-time genomic surveillance on a massive scale. And scientists are using AI to analyze this data, hoping to make more accurate predictions for future vaccines. Finally, there's a growing focus on a One Health approach, which recognizes that human and animal health are linked. By monitoring influenza in animal populations, like birds and pigs, we can better predict and prevent viruses from spilling over into humans in the first place. The global effort to track influenza is one of the greatest, yet quietest, achievements in public health history. For over 7 decades, this worldwide network of scientists has been our sentinel, watching a relentless and ever-changing virus. It provides the intelligence we need for our annual vaccines. It guides our treatments, and it stands as our first line of defense against the next pandemic. The threat from influenza isn't going anywhere. Its ability to change ensures it will always be a challenge. But thanks to this incredible global surveillance system, we don't face that challenge blindly. We face it with knowledge, foresight, and the collective strength of a global community working together to keep us all safe.